and I would like to thank Omics for giving me this wonderful opportunity to present my work in front of you. So I'm Shrawan Baghel from Waterford Institute of Technology, Ireland, and I'm in the second year of my PhD. The title of my PhD is an investigation into the stability and solubility of amorphous solid dispersions of BCS Class II drugs. This research work is carried out in collaboration with Synthesis and Solid State Pharmaceutical Center and Pharmaceutical and Molecular Biotechnology Research Center, and the funding for this project is being granted from Science Foundation Ireland. So first, I would like to give you a brief outline of this project. This project involves 4 different phases. The first one is the crystallization study of the amorphous drug, like how much instability is there in the amorphous drug, what is the tendency to get converted into the crystalline form. The second stage involves the choosing different polymers to stabilize the amorphous drug. In the next phase, we'll move on to manufacture different manufacturing techniques such as hot melt extrusion, spray drying, and supercritical fluid technology. And finally, we'll carry on with the downstream processing of the amorphous solid dispersion products into tablets and capsules. Now, one more thing I would like to say. At this stage, we have finished the first 2 phases of the research, and the second half of the project is on the floor, and hopefully I'll have the opportunity to present it in the next year of the Omics conference. So in the phase 1, we carried out the investigation of the crystallization tendency and kinetics of the amorphous active pharmaceutical ingredients. The model compound chosen for these studies are the dipyridamole and cynarzine. First, I would like to tell you that when you heat an amorphous drug, there are 4 different important events taking place in the DSC thermogram. The first one being the glass transition, followed by the supercooled liquid state, followed by crystallization, and finally the melting of the product. So we first investigated the glass transition event in terms of the fragility. This parameter tells you about the viscosity of the drug around the glass transition temperature and tells us how much it has the capability or how unstable it is and how much it has a tendency to go into the crystalline form. In the next phase, we characterize the supercooled liquid state by using this parameter, glass forming ability, which says that whether the drug will revert back on further heating, whether the drug will revert back to the amorphous state or continue to the crystallization. Then we perform the isothermal crystallization kinetic studies. From this study, we were able to gain the information regarding crystallization mechanism, whether it is nucleation based or diffusion based, and this mechanism is of very much practical importance when you go for the downstream processing of the amorphous based drug products, and I'll show you later in my slides. And in the 4th state, we did the non-isothermal crystallization kinetics, and finally we combined these studies and checked the what impact these studies will have on the stability and whether we can make any predictions regarding the stability of the amorphous drug from first 4 studies. So first, we characterize the fragility, and we have used different techniques to measure this fragility: thermodynamic and dynamic. Dynamic from the extrapolation of configurational entropy, and heating rate dependence of glass transition temperature. Now, there are loads of mathematical equations while characterizing these properties, and I have, just to make it more clear and make my presentation short, I have omitted all those mathematical calculation, but if any one of you is interested, just contact me after the presentation. I'll be happy to share those informations. So I compared these 3 parameters, thermodynamic, fragility, and dynamic, from configurational entropy and glass transition temperatures. And the most relevant one is the heating rate dependence of glass transition temperature. We do get these 2 values as well, but they're not very suitable in our case, and so we rejected these 2, and we established this as the best fragility parameter determinants. And now you can see here, as I've said, that the M stands for the fragility, and it is generally said that if the value of M is less than 100, the drug is fragile, the amorphous drug is fragile, which means that the amorphous drug have a very strong tendency to convert to the crystalline form. And if it is more than 100, it has a strong tendency to remain in the amorphous form, and which makes the formulation much more easier. And the D here is the strength parameter, which is very much similar to the fragility parameter. It again says that if it's less than 10, then the drug is fragile, and if it is more than 10, the drug is strongless. These 2 parameters we get from the Averroes equation, and these are the determinants of the stability or instability of the amorphous drugs. Following this, we calculated the relaxation time to give a more qualitative information regarding the instability, And as you can see here, dipyridamole have a relaxation time of 729 seconds, and cenarizine have a relaxation time of 13 seconds at 25 degrees Celsius. Now, higher the relaxation time, lower the mobility, and if the molecular mobility is very less, it will have a very less tendency to crystallize, or in other words, it will require higher temperature or higher thermal activation to convert into the crystalline form. And these two drugs, I also compared this relaxation time with other unstable drugs such as indomethacin, which has a relaxation time of 1800 seconds, but that is also a very unstable. So dipyridamole is quite unstable, and compared to this, cenarizine is very, very, very unstable. And also, if you can see here, by increasing the temperature 15 degrees Celsius, like under accelerated conditions, the relaxation time decreases for both the drugs by 6 or 7 folds, which quite makes sense because at higher temperature, it will have more thermal input to increase the molecular mobility, and it will require lesser activation energy to convert into crystalline lattice. Now, after characterizing the glass transition event, we move on to the supercooled liquid stage, which is the stage between the glass transition, after glass transition, and before crystallization. Now, what we did here is actually we loaded our amorphous drug in the DSC pans and heated at different heating rates, 1 degree Celsius per minute, 2, 5, 10, and 20. And we measured the crystallization onset temperature and peak temperature, and then we calculated the reduced temperature. This reduced temperature is the ratio of the difference between the melting temperature and crystallization temperature, and to the difference between the crystallization temperature and glass transition temperature. And this gives you information regarding the glass forming ability. Now, the important thing to notice here is that higher the reduced temperature, higher will be the tendency to convert into crystalline form, which we don't want here because it will lead to the decline in solubility, decline in the drug product stability. And as you can see over here, both the drugs have very low, quite high, normally the stable drug will have a T reduced under 0.0001% or something like that, but these two have a quite high, and cenarizine as compared to dipyridamole again shows the high glass forming ability, which means that cenarizine in particular is more unstable. What we also observed here is that T reduced value were found to increase at higher heating rates, which suggests that crystallization tendency for both the model drug increases with an increase in heating rate, and at all the heating rates, cenarizine was found to have lower glass forming ability as compared to the dipyridamole, as you can see. So we can say fragile glasses have poor glass forming ability. Now we move to the third part of our study where we did isothermal crystallization kinetics. Here we loaded our amorphous drug in the DSC pans and at a predetermined temperature, which are shown over here, crystallization temperature, and we check the degree of crystallization over time. And again here we use the Averamy equation, and I have not shown for clarity. If anyone is interested, I'd be happy to share the information. And this is the Averamy constant, and this is the Averamy exponent. Now, the two important observations from this study is the value of N and the activation energy. We have already said that cenarizine is more fragile and have poor glass forming ability compared to dipyridamole, and again it is evident over here. You can see the activation energy under isothermal conditions are lower compared to dipyridamole. Again, these two activation energies are very low as compared to other unstable drug of indomethacin, which has an activation energy of 96 or 97 kilojoules per mole. Another important observation is the value of N. You can see I've shown over in this table that if the value and different values of N will shows different crystallization mechanism. Now, the important thing to look over here is that if the value of N is less than 2.5, it has a high nucleation rate, and if it is more than 2.5, it has a nucleation and diffusion-based mechanism. And the relevance of this is diffusion-based mechanism crystallization requires higher temperature, or it is more stable in simpler words, and nucleation-based crystallization is highly unstable. So we can see over here that cenarizine, the value of N remains more than 2.5 at all the temperatures, which means that it is a predominantly nucleation-based kinetics, and it is very, very, very unstable. And I'll show it later that it crystallizes. Again, we did non-isothermal crystallization kinetic studies using 5 different models as per International Conference of Thermal Analysis of Crystallization Committee recommendation. And what we found that none of the models were actually able to predict the crystallization mechanism very accurately. And so we turned to the model-free kinetics, and we found that the activation energy for dipyridamole is 87 kilojoules per mole, and for cenarizine is 93 kilojoules per mole. And we can see that at different heating rates, you can see the drug follows different crystallization mechanism. It is a shift between the nucleation and diffusion-based mechanism. But for cinnarizine, it is more or less, it first follows the nucleation and remains towards the nucleation-based mechanism. So here we said that both the model compound depends on the heating rate, the crystallization mechanism. And when we did the stability studies, we have shown that cinnarizine is very unstable, it is highly fragile, it has poor glass forming ability and low crystallization activation energy. All these things suggest towards the high instability of the cinnarizine. And when we run the DSC of amorphous solid dispersions of cinnarizine and dipyridamole, we found that dipyridamole remains stable over 2 months of storage, but cinnarizine crystallizes. So as you can see here, also in the XRD data. So all these studies predicted the stability of the amorphous drug and characterize it. So at this stage, we found the need to use a polymer to stabilize our amorphous drug for the further downstream processing. So we move on to the next phase of our study, where we have used different theoretical and experimental investigations for the drug-polymer interaction and miscibility. Now, drug-polymer interaction and miscibility are very important to stabilize the amorphous drug. Higher the interaction and higher the miscibility, more stabilization we will get for the amorphous drug. So this study is again divided into different phases. First is from the solubility parameter approach. This is completely theoretical and based on the group values assigned to different functional groups in the drug and the polymer, and based on the difference between these solubility parameter value, we can say whether drug and polymer will be miscible or not. The second is the drug-polymer binary interaction parameter. This is actually the extension of the Flory-Huggins theory from the melting point depression data. The third is the phase diagram, where we can actually see different zones of stability and instability and how much drug loading we can achieve under different conditions. The fourth one is the drug-polymer interaction in the presence of moisture using dynamic vapor sorption studies. And finally, the role of polymer in maintaining and prolonging drug supersaturation in the aqueous media. So the first thing first, in the solubility parameter approach, I have used two different methods of Tizer and Van Krevelen and Federer method, and average has been taken for the further studies. Now, in theory, it has been said that if the difference between the solubility parameter value of drug and polymer is less than 7, then they are miscible, and if it is more than 10, then they are immiscible. And we found that for both the drugs, dipyridamole and cinnarizine, with two different polymers, PVP and polyacrylic acid, we found the difference is less than 7, which suggests miscibility. Now, we also calculated the interaction parameter at 25 degrees Celsius between drug and polymer, which is found to be quite good between the drug and polymer. And again, this is a completely theoretical approach. So we moved to the experimental side. And here what we did is, to get the melting point depression data, we prepared a physical mixture of amorphous drug and polymer. And it's been said that if you mix a drug and polymer, and if there are more interaction between the drug and polymer, you will get a depression in the melting point, because the polymer will bring down its free energy and bring it towards the crystalline form. So using these equations from the depression melting point data from the DSC, we plotted a graph between this parameter. This is the volume fraction of the drug in the polymer and these parameters, and found the value of interaction parameter is this. Now, the negative value suggests a strong heteronuclear interaction, like a strong interaction between the drug and the polymer. And as you can see, dipyridamole and polyacrylic acid shows the most negative value, which means that it has got the best interaction compared to the other systems. Now, based on that data, we prepared the phase diagram, which has got a solubility curve in red, miscibility curve in green, and the glass transition curve in blue. The significance of this phase diagram is it will help actually the formulation scientists to define the different manufacturing conditions and to predict the stability and instability region. Above this solubility curve, the drug is very miscible in the polymer, and you don't have to worry about the crystallization. It will maintain its solubility and stability. But as you can see, at 25 degrees Celsius, this solubility is very, very low. It is approximately 0.01% weight by weight of the drug loading. So we are actually interested in higher drug loadings. It is this region which is the unstable or metastable, and we have to formulate to stabilize the amorphous drug at this drug loading. So we prepared different drug loading solid dispersions, and the results what we found is, oops, sorry, it's quite not visible. The result what we found is dipyridamole is quite stable with PVP and polyacrylic acid, and cinnarizine is stable only with polyacrylic acid and PVP at 65% drug loading, which falls in this region. Now, the reason why certain drug fractions are stable in the unstable zone and certain drug fractions are unstable in this metastable zone draws our attention to two most important effects. The first one being the anti-plasticization effect. It is the as you can see, this blue line shows the predicted glass transition values from the Gordon-Taylor equation. It is based on the additivity, and using the equation you can predict the TG at different drug loadings of the drug and the polymer. And this red dot shows the experimental value obtained. Now the practical significance of this plotting is if you have a positive deviation from the predictions, it means that heteronuclear interactions are very predominant compared to the homonuclear interactions. In other words, drug and polymer interactions are very much feasible if you have a positive deviation, and if you have a negative deviation, these interactions are very low, and drug will like to interact with its own kind of molecule and crystallize. And this probably explains the stability of these three systems, dipyridamole with both the polymers and cynarizine only with polyacrylic acid. And since we have a negative deviation in case of cynarizine with polyvinylpyrrolidone, that's why we got the batch failure. And then we again investigated the drug-polymer interaction using FTIR, and we found that these three systems shows the good hydrogen bonding interaction between drug and polymer, but only it is this cynarizine and PVP where no interactions has been found, and hence the crystallization. Now, we also investigated the drug-polymer interaction in the presence of moisture at 90% relative humidity under dynamic vapor sorption using ternary flow rehugging theory. And what we found is, as the drug fraction increases, the interaction parameters become less and less negative, which means that the homonuclear interaction predominates the heteronuclear interaction. And finally, it's only for the cynarizine and PVP at 90% RH and at 65% drug loading, we found the positive value, which is again in continuation of the results of the previous works. And in the last phase, we checked the role of polymer in maintaining and prolonging the drug supersaturation in the aqueous media. Here you can see we have loaded 200 micrograms per ml of the dipyridamole and 100 microgram per ml of cynarizine in water without the polymer, and you can see this is the amorphous drug solubility, which suddenly falls to the crystalline drug solubility. But in the presence of PVP, this solubility is quite increased, not for the cynarizine, but for dipyridamole it is quite increased. And for PAA is capable to maintain the approximately all the drug which has been dissolved in the aqueous media in the amorphous phase and maintain and prolong its supersaturation. Now, to quantify this thing, we came up with the supersaturation parameter, which is the ratio of this area to this area. Now, this line here shows the drug solubility in the absence of polymer, which is this actually, and this line shows the drug solubility in the presence of polymer, which is actually this or this. And higher the polymer capacity to increase its supersaturation, this line will shift more and more upwards towards this, which is the hypothetical line where all the drug dissolved remains in the aqueous media as soluble form without undergoing crystallization. So we checked this parameter and found that the dipyridamole and polyacrylic acid performed best, followed by cynarizine and polyacrylic acid. Now the reason for this is the stronger interaction between the dipyridamole and polyacrylic acid. And we can see that in this slide you can also see the in the PVP cynarizine solubility is slightly more than the crystalline form, which can be attributed to the retardation in crystallization due to increase in fluid viscosity surrounding solubilized drug. And finally, I'd like to thank Dr. Niall O'Reilly, my supervisor, and Dr. Helen Fox, my co-supervisor. Thank you for your patience.
Thanks very much, Shrawan. Is there any questions from the floor? Okay. In the interest of time, we'll keep it moving along. Yep. Here's your certificate.